Good afternoon, everyone. It looks like people are just logging in, so I'm going to give it another minute or so everyone can get into the webinar. We'll get started in just a second. I see people are still joining, so I'll give it maybe another 30 seconds and then we'll get started.
I think it's stabilized.
It's stabilized. So let's go ahead and get started. Good afternoon, and thank you guys all for joining this afternoon. I realize many of you might be home with school-age kids on a snow day, so hopefully they're listening also, and we can recruit the next generation of data users. My name is Erin Kaleba. I'm the director of the Data Office for Clinical and Translational Research in the Med School's Office of Research. And in the data office, we're always eager to find new ways to let the research community know about new features in our tools or new services we're offering. And so we have our newsletters, I do presentations, but we wanted to try a new format today and have more of a conversational style. So you could hear directly from one of your colleagues, who I call a super user of the data office tools and services, and what his experiences have been with them. So we are so lucky to have a friend and colleague, Dr. Salim Hayek, from cardiology. So Dr. Hayek's expertise is in cardio-oncology, and he's currently serving as the medical director for Michigan Medicine's Frankel Cardiovascular Center clinics. He has this amazing ability to leverage resources and tools around the university to do incredible research while still being very patient-focused and patient-centric. And his mantra, which I love, is to ensure patients are involved in and fully understand the recommendations and decisions regarding their care. So thank you so much, Dr. Hayek, for joining us. Let's just start. Can you just describe briefly what your broad research interests are related to cardiology?
Of course, Erin, thank you for having me, and hello, everybody. It's a perfect snowy day to be listening into our conversation. Yes. So we hope we can entertain you in a PG fashion. Erin, I'm a clinical investigator, and my expertise is in cardio-oncology, so I see a lot of cancer patients. Mm-hmm. I also do a lot of general cardiology, so I see all sorts of patients with cardiovascular disease. My lab's activity spans a bench to bedside spectrum of research activities. My own expertise is in risk prediction. So I really explore the use of novel biomarkers to be able to better identify who will be at higher risk of having a cardiac event in the future, and also figure out who would benefit more from therapy X versus therapy Y using these novel biomarkers. So this whole concept of precision medicine. And in that vein, I've been a big user of our University of Michigan research infrastructure, including the data office.
That's fantastic. And I think we're so lucky to have a user like you, a researcher like you, but when we first set up the office, our main bread and butter was retrospective research, secondary use of electronic medical record data to either build toward a prospective trial or just do some cohort analysis, things like that. So thinking about a researcher like yourself who covers the full life cycle from basic science to translational science, is helpful for us to see what needs are unmet. So when you think about data office tools like DataDirect and eMERS, or a custom data poll that our SQL analysts have pulled for you, how have you, and at what point during that bench to bedside, at what point of your research were those tools most useful?
Actually, they're useful across that spectrum. So for example, we had been studying the role of a specific protein and its involvement in kidney disease. Mm-hmm. And we had found in certain animal models that that protein was somewhat linked to a certain form of kidney disease in an animal model. So one of the great ways that we use tools like DataDirect and the data office is just to go back to a human population and examine whether the observation that we see in an animal or experimental model validates in a human population. And here at the University of Michigan, because we have the central biorepository, we have this humongous resource of biological samples. Not only do we have the clinical data, you can combine it with biological samples to validate what we find at the level of the bench. Now, as a cardio-oncologist, I do a lot of research in the field of cardio-oncology and how to improve and manage the care of patients with cancer and comorbid cardiovascular disease. And that really requires large amounts of data and large patient population, given how heterogeneous these patients are. And I cannot tell you how amazing it is to have all that data at your fingertips and being able to, again, look at association with outcomes based on for certain medical therapies, even association between biomarkers in that patient population with such ease. And the third aspect that we use it for, beyond just the observational aspect, is in clinical trials. So our lab also runs phase 1 to phase 3 clinical trials. And we use the data office to be able to identify the patient population to recruit. So it really, these tools are useful at every aspect of our research.
That's fantastic. Tell me about your, during the COVID pandemic, we tried to put in place processes to more quickly approve IRB applications and get data to industry and link up those biospecimens with EMR data. Were you looking at COVID against these biomarkers for cardiovascular disease, or what was because I was impressed how rapidly you were able to get some really meaningful research findings on COVID?
So at the early onset of the pandemic, back in March 2020, we designed the Michigan Medicine COVID-19 cohort, or M2C2. And this was meant to be a prospective collection of biological samples and clinical data from all patients hospitalized specific for COVID-19. And really, our data collection was supplemented by the self-serve tools that we had here at the university, such as DataDirect, allowing us to extract, discrete data. For example, the labs, the medication, vital signs, things that are discrete. But I'll tell you, one of that endeavor also highlighted some of the challenges of using these types of data pools, because one of the major issues that we encountered in COVID research in general is that a patient hospitalized with a positive COVID-19 test does not necessarily mean that they were hospitalized for COVID-19. So a human element in reviewing the chart and adjudicating who was hospitalized for COVID versus, with a positive SARS-CoV-2 test was absolutely necessary. Nevertheless, we managed to build this cohort and are still collecting data, and we could not have done it at that scale of now. We have about 4,000 patients without the use of adjunct tools such as DataDirect.
Yeah, that's fantastic. So I wonder if we'll ever get to the point, and we can think about this toward the end of this topic, but where the tools will be so good you wouldn't need to put human eyes on the record. And I just don't know for certain. There's so much that's context based that I don't know that the tool will ever be able to discern well, this was a hospitalization complicated by COVID, but not because of COVID, things like that. We might always need that human component. But tools like eMERS allow you to put eyes on the actual medical record notes and things in a more efficient way. But I don't know that we'll ever get away from that, from actually having the human interpretation in there.
Now you bring up a very important point. So I'll give you how eMERS helps. So for example, we did one study on metastatic melanoma. It's a cancer of the skin, and we wanted to figure out which patients had metastatic melanoma to the heart. And I mean, we have thousands and thousands of metastatic melanoma patients. It's impossible to review every single chart. So that's where we used eMERS. We used eMERS to search through the text. So we used DataDirect to pull the patients with melanoma. Then we combine that with eMERS to look through the text and identify any type of wording that would indicate that the patients might have metastasis to the heart. And that's when you shrunk from 10,000 records to maybe 100 records, and these are easily reviewable. And so, and that is one of the ways that you could use these tools to make your job much easier. And even if you absolutely needed the human input, you can minimize the effort to that certain subset.
Just for those who are listening in, I did not give Dr. Hayek any of these answers. He really is such an amazing user of these tools and pushes us to be better with them. Do you have a team that helps you identify your cohorts, pull them from DataDirect into eMERS, do all of that? I mean, how much is you versus, some fellows, some grad students, some med students?
So I'm very hands-on when it comes to
data. That's what I
thought. And you can tell from my DataDirect tools. I love data. I actually, one of the parts I enjoy the most about my work is working with data, and cleaning data, understanding the fallacies of data. And that's why I'm very skeptical of a lot of stuff that gets published out there, because I understand how data can point you in the wrong direction if not used well. But also it's one of the reasons I appreciate our research infrastructure and what the data office has done. But I do the vast majority of my work myself, and I train some of our team members to do recurrent pulls depending on the scenario. But when it comes to the initial data pull, I'm the one on it. Not that I would recommend everybody to do that. I think if you find the expertise or find the help and to do it, but it's crucial to understand, to look at the data, know what it is in order to make the appropriate inferences when you're using it.
Right, absolutely, especially when you're talking data that was collected to deliver clinical care. It wasn't collected for research. So it has, there are caveats to what you're able to say about it. But so you give such great feedback about how the tools, the people, the services have augmented your research. We're very lucky with DataDirect and the data office team to be small enough and nimble enough that we can react to suggestions or corrections. So I'd love to shift now to, if you have any thoughts around where the tools could improve, how we can start to plan for, this ever increasing amount of data that's out there. So you and I have talked about things like starting populations or computable phenotypes.
Right.
So if everyone is studying melanoma may be a bad example because maybe there's only 2 or 3 codes for it. But if you're studying something like heart failure, there are so many ways to define it. Is there a nationally validated, agreed upon definition that we could build into the tools so that you could go in and grab heart failure plus COVID plus this? So I'd love to hear if you have any ideas about where we should go next. We're at a point where we're maintaining a lot of what we built, but we're ready to be innovative and push ourselves to be responsive to the next type of research.
Right, that is truly the next step in advancing our services, computable phenotypes, because again, one of the biggest barriers, I would say, once you get the data is sifting out who truly has the phenotype that you're looking for or the disease that you're looking for, since ICD codes are completely unreliable. And anyone who does clinical work knows that. Simply because of the virtue of how we use those. We use them for billing. We don't use them truly for diagnosis. So we will bill using an ICD code, even if the patient was not diagnosed, but because we're suspecting it. So ICD codes alone are completely fallible. And I would be very skeptical of any research that's solely focused on ICD codes, unless, again, there's millions and millions of patients to compensate for the error rate in the description of these patient population. So devising strategies to refine these definition is important. And there's a lot of work surrounding that across, universally, because this is a problem not just with our system, but everywhere. And there are algorithms being developed. And I believe that the best would be a combination of natural language processing, ICD code, as well as lab work. So a combination of discrete variables and text variables. And eventually you're not going to be able to, unless reviewed manually, to 100% guarantee that that's accurate. But if you create a sort of confidence scale, so a confidence scale that patient has maybe, the odds that this is a correct diagnosis of heart failure is 70% based on X algorithm, would be tremendously useful for anyone doing large data research. And I believe that's the next step, not really creating something that will guarantee you the diagnosis, but rather a metric based on a computable phenotype with a percentage confidence.
Right. And then I can imagine you would say in your paper, we know we were too inclusive. And so we have patients that probably don't, or we were very narrow. So we know we missed patients, and a little bit of that sensitivity and specificity to, no one would expect you to get it perfect, but to be able to actually speak to where you might be erring and is the tent too big or too small would be really powerful.
Right.
So you and I have had the opportunity to review tools like DataDirect, but that are more national in scope. So one is called Trinetics, that a group of us were asked to review and consider. And that would be from other academic centers that have also agreed to contribute their data. And U of M has not agreed to that yet, but we're testing it out. There's also Epic itself is looking to host a community of Epic users to give de-identified data to have a much larger N, finding especially rare patients with rare diseases, but also be able to build more of those models that really take a large number of patients. So the nice thing about being able to review these other tools is we could say, oh, I love that feature. I love that feature. Maybe we could do that in DataDirect. But what are your thoughts on the advantages of some of these third-party tools that have data beyond just Michigan medicine patients for you to look at?
Let's take a step back here. If you ask anybody, whether a data scientist or a clinician, what is the next big step in precision medicine? It's all about sharing data.
Yeah.
And that is the only way we're going to be able to overcome a lot of the limitations of, single center analyses, selection biases, looking for essentially the right phenotype to be able to treat appropriately. You need very, very large sample size. For genetic analyses, omics, proteomics, metabolomics, all of these technologies rely on large sample size and appropriate selection of the patient population. The future is in data sharing. The future of science and medical science is in data sharing. So what Trinetics is doing is really putting this into implementation. So it is commendable. They've now, I believe, maybe 40 institutions. If maybe correct me if I'm wrong.
I think that's right. 40, I think it's around 40. Like yeah.
So I was truly impressed at what they've done, and I believe, regardless of the return on investment, etc., let's say just ideally and morally, all institutions should contribute, because this will lead to the advancement of healthcare, right, as a whole, as a big picture. in their current state, what we've tried out, you and me, is really something similar to DataDirect in some ways, which really speaks a lot to how amazing DataDirect is. So they've emulated a lot of what we do. Here, I think our tools are still a bit more advanced, but they've made theirs compatible to include 37 institutions, which is amazing. And if you ask me what we should do, we certainly should contribute, again, as a matter of principle. And if contributing opens up access to our investigators to this tremendously large data set, then that will advance our own science and a science for everybody contributing that much more.
Yeah, and I like to think about ways to, it didn't seem, in the past when we've been part of things like PCORI, other national opportunities to share data with our partners, there was a tremendous amount of upfront work to map our data elements to a common standard. And I think we're seeing tools now that can build a little more adjustment on the tools end so that you don't have to spend 6 months of in-depth mapping of data elements. Hopefully, I don't know, we could learn that's not the case, but.
Now, I just wanted to point out that they're complementary, TriNetX and DataDirect. So it's not one over the other.
Yeah, talk about that, because DataDirect is also interfaced with the central, biorepository and the precision health, genetic info and increasingly some wearables data sets. But how else do you think of them as complementary?
So remember what I mentioned earlier, DataDirect allows us to create new cohort and complement the data from that building of new cohorts, right? From our own patients that we see in Michigan Medicine. TriNetX is purely secondary data analysis, right? So it would allow us perhaps to help plan for recruitment, just similar to what DataDirect does, again, at a much larger scale, which if you think about clinical recruitment, then you're planning for other sites too. But again, DataDirect allows us to do the secondary analysis in addition to the development of creation of new cohort, which is not what we can do with TriNetX. And so that's why they are complementary. TriNetX would be great because you can validate any of the findings that we derive from our own populations here.
Right, and I think we know that, and even in the way NIH wants to fund networks rather than individual centers, a lot of time you take Michigan Medicine, we're not nationally representative of ethnic and racial and other demographic characteristics. And so our findings are appropriate for our population, but they're not nationally representative of what a lot of other patients in other parts of the country look like. And so I do think the power too in having a larger, more diverse patient population like you said, to validate, to test our own findings against is really powerful, because you can never expect one health center to look representative of the US or the world for that matter. So because you look at predictive algorithms, what are the key factors about a patient, do you think, that'll be our next type of data we go after? So we're getting better about patient-reported outcome surveys. We're getting better about collecting data from a patient when they're not in your clinic for 15 minutes, whether it be through an Apple Watch or a Holter monitor, or and we know about where they live, some geolocation and SES data. What's missing that would say, I could predict that this group will get this disease and this one won't, maybe that we're not collecting right now? Do you have any ideas on that?
Low-hanging fruit, Erin, is our own imaging data. I know we're having some progress, right? With X-rays, but we have a wealth of imaging data, and it is about time that we translate those into data that's usable for the scientific community. So I think that's a low-hanging fruit there. It takes some work, but it is doable. And one of the very important parts of a patient assessment that is difficult to code is really the frailty or exercise tolerance, or just general physical ability of a patient. And that's really difficult to capture in a clinic setting unless you're being seen by sports medicine or physical therapy. And so that's where wearables come in. The problem with wearables is that not everybody's wearing them, right? It's a certain subgroup of patients who are usually very motivated who are wearing these. But I know, again, there's a lot of our colleagues who are working on collecting wearable data and integrating it in DataDirect. But again, as a top reason, I would say imaging.
Great.
The third is getting access to our patients' data within healthcare systems in Michigan.
Yeah.
And whether that's possible, the legalities around it, I don't fully understand. But whatever we get in care everywhere, that's a wealth of data that can supplement our own and give a better perspective of the patient's course once they leave our institution, which is
right.
the our patients, right? They leave, they go back to their communities, and that data we don't have access to, at least not through an easily downloadable method.
Yeah, that's the blessing and the curse of being a tertiary care center. Is some come for one surgery, but then receive the rest of their care closer to home. And there's claims data, but that's really about health services research, which is very powerful to look at utilization. But I know what you're saying about some of the more granular healthcare data from outside institutions. I think it'll be interesting to see what the trend continues to be in terms of patients signing a universal consent to say, I'm open to you accessing all of my healthcare data, not just the data at this institution, and how I think we'll see data aggregators come about that will help do that for patients who do sign a broad consent. Again, that might not be a representative patient population who's open to that, but that's one way to get around some of the legal hurdles about how we can use outside data.
Or an opt-out. I don't know if an opt-out.
Or not.
These discussions have happened, but maybe
we could go a couple more hours if we got into the opt-in, opt-out, I think. Some of our researchers are curious how to get started partnering with industry. And we have Fast Forward Medical Innovation and Innovation Partnerships, which are such incredible business development resources. How did you get started? Was it you seeking out someone who had an assay or a development you wanted, or does industry come to you? We often get asked, how could a researcher get started?
So there are two ways. The way that perhaps we went through our lab is that we built resources. And so when you build resources, just as the Michigan Medicine COVID-19 cohort, the Emory Cardiovascular Biobank, our now Care BMT cohort here at the University of Michigan. So when you build these resources, and you measure novel biomarkers, et cetera, you will inevitably attract attention from different people who want to leverage these resources. Yeah. So that's, I think, the way that, that maybe takes a bit more time and effort. But, what I have found my interactions with industry, they are very willing to help and need help, and they often lack perspective, and they welcome it from physicians and scientists. And I have never found a company to be reluctant to hear me out or listen to a proposition or even fund. I mean, have been willing to support in one way or another. Not just companies that have approached us, but the ones that we have approached. For example, if they were working on a product that we were interested in or a disease state that we're interested in, we're always ready to listen. And if they haven't reached out to you, is it because they may just not know about you, but they want to know and they welcome it. And I find that industry funding really helps create new resources and allows you as an investigator through that funding to perform high risk, high reward research that is sometimes difficult to fund through NIH and other venues.
Is the turnaround time of getting industry funding part of what makes you able to create new compared to the slower NIH, or is it just that you can be a little bit more creative in what you're suggesting you'll you'll develop or study with the industry?
Certainly. So the timeline aspect is huge, right? NIH is really year over year. A grant proposal goes in, gets reviewed, gets scored, needs to be resubmitted. I mean, it's a year over year thing, right? While with industry, it's not a matter of rejection or acceptance. They will often sit and discuss with you what needs to be refined, how they can help you improve things. There's also a level of flexibility in the research and what you can propose. And as anyone who has submitted to NIH knows, right, there's not just about how good your science is, but it's about on whose lap that proposal falls.
Correct.
If it falls in the hands of a competitor or someone who doesn't know your science very well, it might be the best science in the world. It will end up triaged. So again, NIH research and industry funding are complementary. I think it's to do different things. And we collaborate. We have funding from both NIH, from industry, from foundations, and we do very different things with these sources of funding.
I think diversifying the funding streams is the most powerful, as you said.
Agreed.
I am going to open up the question and answer. If you all have questions for Dr. Hayek, just type them into the Q&A and I'll read them aloud. I have to ask before I get started with the Q&A questions, since you've been here almost 4 years, 5 years almost, from Atlanta, and we're in the middle of a blizzard, what's your favorite thing about Ann Arbor? Since you
I was going to say the snow, but nobody's going to believe me.
No one's going to believe you.
Actually, with the snow when it first falls, no matter, it's always, I just look at it with wonder every single year.
It's gorgeous.
After a few days, I'm sick of it. But that first snowfall is just wonderful. I'll tell you honestly, what I love about Ann Arbor is its people. I mean, it's, how often do you live in a town where your neighbor could be, Nobel Prize nominee or a winner, or wearing sweatpants, working the yard, and end up being some sort of super expert in their field, or when you're at the grocery store, you bump into ex-scientist or professor and strike up interesting conversations. I mean, this is really a bubble. It's an amazing bubble with a lot of interesting, geeky people, and I love it.
I do too. I love it. I can't go to Target looking too terribly because I'll run into someone I know inevitably, but it's always, I love that aspect too.
But what? I'll tell you that regarding that comment is that, it's also a town in which you go as you are. So even if I wearing sweatpants, I'm going to find my boss wearing sweatpants too at the same time.
It's true.
And I love it. I love it about this. You wouldn't call me, dead wearing sweatpants in Atlanta, but here it's fine.
I know. My brother lives in Southern California, and he misses the casual Midwest because everyone there is waiting to be discovered for a movie. I have two questions that have already been posted. One is from my colleague in IRB, Lark, and she says, does NIH, all of us, count as a similar platform? So Lark, I think you're probably talking about when we were discussing tools that are available that aggregate data from national sources, could we consider the NIH, all of us program as one of those? And I would think that would be considered. Dr. Hayek, do you agree that the precision health platform, all of us?
Right. I mean, all of us is a cohort that's being built independently, right, involving a very large number of centers. So it's a bit different as what Trinetics is trying to do, which is incorporating secondary data or data from a lot of different institutions that we can combine with. Now, the All of Us platform will be made available for other users, right? So it's just a bit of a different type of platform, different type of data than what's available from Trinetics or DataDirect.
Agree. And I think, Lark, what I like about your question is it's getting to this, Dr. Hayek said that the future of science is data sharing. And so we're going to see lots of different types, but NIH has really tried to get, genetic and other characteristics of data shared nationally to try and reach critical mass 4 different discoveries and advances. So there is part of that picture of national data sharing.
Correct.
And then my colleague Jasmine wisely pointed out that the FEKB database, if some of you don't know, this is out of Vanderbilt. It's called the Emerge Network. And there's, I'm not sure now, I'm going to say almost 100 computable phenotype definitions that have been submitted by different members of this network. And so you can see what Northwestern's, myocardial infarction phenotype is compared to Duke's. So it's even within one disease or condition category, there will be multiple definitions. And it's an incredible resource. If the others haven't looked at it, we often, when an investigator at Michigan suggests a new phenotype, like we just had one come out of gastroenterology, they were interested in Crohn's disease and IBD. So the first place we go is this resource called FEKB, because it's likely someone's already thought about what codes, what phrases or text from an NLP perspective really say this patient has Crohn's or IBD. So I hope all of you could see that link in the Q&A, because that's really helpful. Dr. Hayek, have you looked at that resource at all?
Yes, we have, and that's part of what we've proposed for NIH for a study that we hope to then integrate in DataDirect using FEKB. But it's a starting point, again, because this needs to be validated, et cetera, and within our own system.
That's true. Yeah.
And there are others, other approaches, but FEKB is an important one.
And I know the last time we did an assessment, about 75% of the algorithms in or the phenotype definitions in FEKB required text, which is not surprising. There's very few conditions I think you can reliably define with codes, discrete codes alone. But yeah, thank you, Jasmine, for pointing that out. Someone else asked, in terms of getting started with the tools, with eMERS, with DataDirect, with things like that, is your recommendation just to jump in and start playing with them? Since you're hands-on, I don't know if you watched hours of videos on how to use them. Do you just jump in and kind of start playing with the functionality?
Exactly. I would recommend before watching any videos, just spend some time clicking through doing it yourself. DataDirect is very, very self-explanatory. There are certain quirks, and and once you end up reviewing the videos after familiarizing yourself, you can also, the staff has been tremendously helpful. You can ask them questions, they respond in a very timely fashion. But the best way is to start by exploring it yourself, then build on that by reviewing the videos, because they will answer some of the questions that you will get as you're using the tool. I think the biggest the hurdle is once you extract that data, and it's often a lot of data, understanding how to combine it. And often what will happen is that you may extract the data and realize that's not what you're looking for, and then go back and fiddle around with it till you learn how to exactly choose the right variables and definition. But it is certainly something that any non-coder can do. I do not code. I have no idea how to code. But so this is, and I can use DataDirect to its entire capacity. One thing I wanted to point out regarding that is I don't know if people appreciate how important, how amazing it is to have DataDirect. And other institutions, getting our hands on data like this involves, coders, paying thousands of dollars of hourly work to extract a data set and then look at it and say, that's not what I need. And then wait another 3 months. Well, you can do that within minutes. I mean, it's just incredible. And I'm happy to personally walk through anybody who would like to use this or who has questions.
I did not pay him to say that, you all. He's really that generous. Yeah. I think you've probably learned a lot of what not to do and some tricks just from your use of it. I have one final question before we sign off. My colleague Lisa Day has asked, do you suspect some institutions will hesitate to collaborate and share data without patient consent?
So Erin, you probably have a more detailed answer than I do. And correct me if I'm wrong. You've been in these discussions more at the institutional level more than I do. I think it's less. It may not be as much about patient consent, but maybe more about the perspective that data is money these days, and because we can de-identify data very, very safely and permanently. So I doubt that it's a matter of breach of confidentiality or legal issues, and it may be a matter of, again, who's going to benefit and why, and who will own the results. So again, Erin, you probably know more than this than I do.
I mean, that's exactly what the research suggests, is that if patients know that their data are being used to improve quality to research, they're okay with it being de-identified and shared. If they think it's landing in the hands of insurance specifically, some research has found no, and some industry no, without them being aware of it. But that's, I mean, it's the balance that we try and strike is to say, our patients have benefited from decades of research, and the vast majority of our patients want to pay that forward and continue to contribute to advances going forward. But who benefits from it, who's going to make money off your data, that's where it gets tricky. If it's for researchers and scientists, patients are usually very happy to contribute their data. So that's what we know so far. All right, anonymous attendee, great fireside chat on a snowy day. Well, great. If there are no other questions, I think I'll end the webinar here, but please reach out to me or to Salim and let us know. If you like this format, let me know. We're continuing to find new ways, but I just thought it was so valuable to hear from someone who's actually rolled up his sleeves and used the data and used our services and keeps us improving at all times. So Salim, thank you so much for joining me today, and I look forward to continuing to work with you.
Pleasure is mine. Thank you, everybody, for joining.
Thanks, guys.